Sorry, I left my glasses at home, so I'm probably going to be lecturing on my son's because they're prescription. So apologies in advance, but I can't really read the screen. Come on, you potato. Cool. All right, well, let's get started. So, good morning, everyone. Hope you all had a great weekend. So today we're going to continue our neuropharmacology lecture series. We're moving on from kind of mental health disorders and their treatments. Today we're going to talk more about a neurological disease or disorder, specifically a neurodegenerative disease, which is kind of the blanket kind of symptoms associated with dementia. We will focus a bit more on Alzheimer's rather than dementia, just because it has some very kind of characteristic mechanisms. And yes, we'll go on to the neuropharmacology of that. So what do I want you to get out of this lecture? So similarly, the running theme over the past few lectures is that even though it's not a neuroscience unit, I still want you to understand some of the pathophysiology of dementias and Alzheimer's disease. Mostly we're going to talk about Alzheimer's disease, so we'll focus on amyloid beta containing plaques and the neurofibrillary tangles. I don't know if you've heard of those before, but we'll briefly touch over them and why they're important in the disease. And then after we talk about that, we'll then get into the neuropharmacology, specifically focusing on common pharmacological treatments of dementia and their mechanism of action. We're mostly going to talk in the context of cholinergic drugs, so drugs that affect acetylcholine neurotransmitters, which is very important for things like memory and mood regulation. We'll touch briefly on serotonergic drugs again. So we talked about SSRIs in the context of mood disorders. We'll briefly touch on them again for dementia as well. And then we'll move on to some more kind of alternative treatments for dementia and Alzheimer's, mostly focusing on antibodies that are specific to amyloid beta. And then moving on, I've just got one or two more slides just on kind of like a non-pharmacological treatment, and that's going to be focused on ultrasound stimulation that we briefly touched upon in one lecture. So first of all, what is dementia? I'm sure everyone here is at least familiar with some of the symptoms of dementia, but it's an umbrella term for several neurological conditions. So Alzheimer's falls under it, frontotemporal dementia, even some forms of Parkinson's disease can fall under dementia as well. And this is just taken from the Dementia Australian website, which is a really great resource if you want to learn more about dementia and kind of how it affects people, society, and some treatments as well. But mostly what we classify as dementia is when you see deterioration in more than one cognitive ability, and this is below premorbid levels that are severe enough to impair daily function. So what does it mean? Usually it means that there's a very strong deterioration in cognitive abilities that can't be attributed to things like developmental disorders or something going wrong with development or even during the embryonic development as well. It's usually something that is acquired over time and can't be attributed to those specific conditions. Again, that's differentiated for developmental disorders where something might be going wrong during development which can cause a disease. So it's just, there is a little distinction between kind of acquired diseases like dementia compared to developmental disorders. So what kind of symptoms do we see? Well, the biggest symptom that we see is memory loss. So this can be very like non-severe, so just things like, you might forget your keys or you might forget where your house is, to more kind of severe forms of kind of memory impairments where you might forget who you are, you might forget who your loved ones are, things like that. So it's usually termed around memory disorders, but it can also, memory impairments, but it can also be linked with mood dysregulation as well. So you see a lot of people with dementia, they might have inability to regulate their emotions. They can go from depressive states to manic states, even becoming very agitated very quickly. So it's not just memory that's affected, it's multiple cognitive systems. And we can classify dementias into different types. So you can have primary degenerative dementias. This is where you get neuronal loss in very specific and functionally connected brain regions located with the disease. So you usually see this with dementia and Parkinson's disease-like symptoms. So you usually get kind of neuronal loss in very specific regions of the brain, like the hippocampus, and even some motor pathways as well, so things like the basal ganglia. But then you can also have secondary kind of dementias as well. So usually these secondary or symptomatic dementias, these usually result as a response to something else. So it's not just really the neurons are dying, which is causing the disease. The neurons might be dying because another disorder or injury caused that disease. So you can have you can get dementia in cases where you have disorders that directly damage brain tissue, so traumatic brain injury, so that's the secondary case. So the primary injury is the traumatic brain injury, but then secondarily you might get dementia associated with it. So it wasn't kind of what's generating the neuronal loss in the primary degeneration, but it's associated with a secondary mechanism. You can get disorders that increase intracranial pressure. So if you have like a stroke and you have blood accumulating in the brain, that can cause damage to brain regions, and if those regions are important for memory, they can affect that as well. So just know that you can get kind of primary pathogenesis of the disease versus kind of like a secondary mechanism as well. And breaking these down further, so here's actually a list of many different primary degenerative dementias. As you can see, there's a lot there. So some of the kind of more common ones that you're probably associated with or that pop up a lot is Alzheimer's disease, which we're going to talk a little bit about soon. Frontotemporal dementia again, or which you might hear in regards to Parkinson's disease as well. Parkinson's disease dementia, frontotemporal dementia with motor neuron disease, Huntington's disease. So a lot of these kind of fall into not just, damage to, specific neurons in the memory system, but a lot of them are associated with other other neurodegenerative disorders that target the motor system as well. So Parkinson's disease, Huntington's disease, motor neuron disease as well. And then secondary dementias, again, these can arise from many different kind of sources. Some of these, again, like I mentioned, is stroke, demyelinating disorders like multiple sclerosis, traumatic brain injury, glioblastomas, and then even toxins or even some specific drugs can lead to dementia as well. So there's a whole range of different pathophysiologies. So not every dementia is exactly the same. There's usually people experience very different symptoms, usually similar symptoms, but kind of can be different and in different brain regions as well. So just trying to just reiterate again, it's a very complex disease, much like the other mental health disorders that we've been talking about these past few days. So what are some actual brain changes that we see in the brain? Again, you don't need to focus on this too much. It's just trying so you can understand in your mind what's actually happening in the brain. So I don't know if you've seen any of these pictures before, but one of the biggest kind of changes that we see in the brain is that you see lots of macroscopic brain changes. So this is just looking at the level, the non-microscopic level, so things that you can see with your naked eye, pretty much. So if we obtain the brain of someone that has Alzheimer's disease versus someone that doesn't have Alzheimer's disease, one of the things you see is brain atrophy. So you do see some changes in the brain where there's loss of brain tissue. That's one of the big hallmarks of the disease. So what are some of the changes that that you can see that kind of indicate that there's brain atrophy? Well, you see widening of the sulci and gyri spaces. Don't worry too much about what sulci and gyri are. Basically, they're just these little kind of ridges in between the brain tissue. And as you can see in someone that has severe Alzheimer's, you see kind of like a widening of those sulci and gyri, so they get bigger because basically there's loss of the brain tissue next to it. So again, just reiterating that you're losing that brain tissue. Enlargement of the frontal and temporal horns of the lateral ventricle. So I mentioned ventricles before. They're these kind of canals that run through your brain that are really important for insulating the brain. And those actually get bigger because you're losing the brain tissue around it, so those chambers get bigger. And I think that's what's showing you here. So you can see this ventricle around here has gotten bigger compared to this one. And another change that you see is loss of pigmented neurons in the locus ceruleus. So the locus ceruleus is shown here and in this picture. It's a brainstem structure, and there's a population of norandrogenic neurons there that are really dark in color. So you can see this little dark spot in that white circle. But in this brain that had Alzheimer's, you don't see those pigmented neurons in that brainstem. So you're losing those neurons. These features, again, they're common in all forms of dementia, and depending on kind of the severity and progression of the disease, you might see differences compared to different people. But these are some of the macroscopic, you see them under the naked eye, changes in the brain. But we also see some microscopic changes that lead to these macroscopic changes. And when we're talking about dementias and specifically Alzheimer's, we usually see two hallmark kind of symptoms of the disease. And that is the accumulation of amyloid beta containing plaques and also neurofibrillary tangles, okay? So let's unpack these. What actually are these mechanisms or symptoms? So amyloid beta is a protein that's found in neurons, okay? So seemingly some neurons seem to just have this amyloid beta protein. Excuse me. But in dementia, you start to see that these amyloid beta proteins aggregate a lot, okay? So they seem to become a bit sticky, they stick together, they get a little bit bigger, and this can actually be neurotoxic. So the more that they build up, the more toxic it becomes to the neurons. This can cause their death through things like reactive oxygen species, caspase activation, apoptosis, things like that. And it's mostly the postsynaptic compartment of neurons that's really sensitive to this amyloid beta accumulation. So the postsynaptic compartment, we're talking about things like receptors for glutamate receptors, so specifically metabotropic glutamate receptors and NMDA receptors. They're very sensitive to amyloid beta toxicity, and they seem to degenerate to this aggregation. So you get an alteration in kind of that excitatory neurotransmission between cells because you lose these receptors and these neurons then begin to die off. And this is just a picture showing you neurons with amyloid beta accumulation. So the green is showing you accumulation of amyloid beta. Red is showing you just MAP protein. So this is just a straight up microtubule protein. So it's just showing you the neuron. And then when you merge them, you can see this aggregation of amyloid beta. And what's really interesting is that, so you usually see more amyloid beta accumulation with dementia patients. But this is just highlighting again that we just see amyloid beta in our neurons anyway. And we're not really sure what causes the accumulation of amyloid beta, specifically in dementia, and why it can be neurotoxic to dementia patients, but then not neurotoxic to other people that don't have dementia, because it's always there. And one of the big kind of goals of figuring out what causes dementia is kind of understanding this amyloid beta aggregation. But what else do we see? Well, we see changes in tau pathology. So tau is, again, another protein. It's mostly associated with the cytoskeleton, so it's really important in the structure of neurons. We see it in normal neurons everywhere. They express this tau because it helps just with their structure. But what happens in dementia is that this tau becomes a little bit agitated and it starts aggregating on itself, similar to the amyloid beta. So it starts taking this abnormal morphology and becomes these tangles, and these tangles can also be neurotoxic to cells as well. And this is just showing you tau in a neuron as well. So all this is a grayscale image, so where you see these kind of white hotspots, this is just showing you tau. So just going to show you that all neurons seem to express tau, but in dementia, something happens to this tau where they become a little bit more aggregated, and that accumulation can be neurotoxic as well. So two kind of characteristic mechanisms that we see. So amyloid beta aggregation and neurofibrillary tangles by this tau aggregation as well. Don't worry too much about this, but this is, in case you're interested, is that we still don't, again, we don't really know what causes dementia. So we see in dementia patients accumulation of beta amyloid, and we see neurofibrillary tangles. But it's kind of that chicken or the egg kind of paradigm. Do people with dementia get dementia because they have beta amyloid and tau accumulation? Or is kind of something else going wrong? Was it already present before they had dementia? And we don't really know. Because some people actually think that this beta amyloid accumulation and tau aggregation might be the body's response to kind of curb the symptoms of the disease. Yes, I was listening to Mr. Vane. Oh, it's not showing up. Okay. And this is something we'll talk about with the amyloid beta antibodies, but maybe these proteins are actually your body's way of fighting off the neurodegeneration, but we don't really know. But just letting you know that, again, that we still don't know why they can be why these hallmarks might be neurotoxic. So for example, one hypothesis suggests that amyloid beta first gets accumulated. This causes phosphorylation of tau, which then is toxic to cells. There's other hypotheses that suggest that amyloid beta is toxic to cells, but how toxic they are is dependent on tau. And then there's other hypotheses that suggest that amyloid beta and tau both work on mitochondria, and this increases reactive oxygen species to kill cells. So don't worry too much about the mechanisms. I'm just going to show you that these are just some specific hypotheses that suggest why we might see toxicity, but again, we don't really know the full story there. So how do we manage dementia symptoms? And again, manage is underlined here because mostly a lot of our treatments, until recently, have always been about managing the dementia, mostly managing the neurological degeneration. We can't really stop the degeneration, or we can prevent the degeneration, but we can't, increase kind of the healing afterwards, and we can't replace lost neurons. So we can target neurodegeneration to prevent those amyloid beta and neurofibrillary tangles to kind of slow down the loss of neurons. But mostly a lot of the treatments work on managing the behavioral and psychological treatments. So again, in patients that have dementia, you can have a lot of neuropsychiatric disturbances alongside this memory impairment. So you see psychosis in a lot of patients. You see increased agitation, increased aggression, increased irritability. You see a lot of kind of depression and anxiety as well. And sometimes these behavior and psychological symptoms can actually appear before the memory impairment as well. So you might notice that, like anecdotally, maybe an older family member is just all of a sudden becoming a bit more anxious than usual. Sometimes that could be associated with dementia as well. I'm not saying it is, but it can in some cases. And this is just showing you a breakdown of some of these behavioral and psychological symptoms and how prevalent they are in people with dementia. So you can see in this case in people that are diagnosed with Alzheimer's disease, and again, you can only be diagnosed with Alzheimer's disease post-mortem, but of the people that have Alzheimer's disease, about 30% of them display psychosis towards the end stages, 10 to 20% experience hallucinations, and up to 40% with delusions. So there's a lot of these strong neuropsychiatric disturbances that occurs with a lot of these dementias. So a lot of treatments work to kind of stem those symptoms. So what are some treatments that we can use to ameliorate those symptoms? So one that we haven't talked about yet, we kind of touched on it a little bit in one of the previous lectures with neurotransmitters, but we can actually look to the cholinergic system. So what is the cholinergic system? Mostly this is focused on the neurotransmitter acetylcholine, okay? So we've talked about neurotransmitters, glutamate, which is excitatory, GABA, which is inhibitory. We've talked about monoamines like dopamine, serotonin, noradrenaline. Well, acetylcholine is its own neurotransmitter. It has its own receptors in the brain and throughout the body. And so a lot of these kind of acetylcholine receptors and neurotransmitters are important for some of these dementia symptoms as well. And this is just a cartoon showing you some of the acetylcholine in action. So this is a presynaptic neuron, a synapse, postsynaptic neuron. So when there's an action potential, neurons that have acetylcholine will release it into the synapse, and that can bind to their postsynaptic receptors. Now there's two different subtypes of receptors for acetylcholine. You have nicotinic receptors and also muscarinic receptors. Nicotinic are more ion channel based, whereas muscarinic are more metabotropic based, so similar to a lot of the other systems we're talking about. But acetylcholine also has its own enzyme that breaks it down, and this is it here, acetylcholine esterase, or I call it ACE in my head, but it's ACHE. So this enzyme breaks down acetylcholine so that this choline gets reuptaken and then produced back into acetylcholine. So this enzyme just breaks it down so there's less acetylcholine in the synapse. So similar to what we talked about with the monoamines as well. And really interestingly, we see a lot of loss in cholinergic neurons in some different forms of dementia. So specifically in Alzheimer's disease, we see a loss in cholinergic neurons. Now, acetylcholine neurons are present throughout the brain. I haven't showed you a heat map here, but it's very similar to kind of serotonin that we talked about. A lot of them are focused in a lot of reward and mood regulation pathways as well. So we're kind of affecting those similar systems. And there's a variety of different acetylcholine or cholinergic drugs that are used in dementia. So two we're going to talk about is these two here, so donepezil and rivastigmine. And they're all made by different companies. Some of them might be a bit more neuroprotective, so they might be able to protect neurons more than others, whereas others can actually manage the symptoms a bit more. But there's a lot of kind of unknowns with these drugs as well, because there's not a lot of information on some of these mechanisms, but there is some evidence for them. So the first one is donepezil. I'm probably pronouncing it completely wrong. This is its structure here, but this is a reversible acetylcholine esterase inhibitor. So it's inhibiting this enzyme that breaks down acetylcholine, okay? So if you inhibit the enzyme that breaks down acetylcholine, there's going to be more acetylcholine present in the synapse because it's not getting broken down, okay? And this actually provides symptomatic relief of Alzheimer's disease symptoms. Mostly it increases or improves memory and cognition relative to placebo. So it does affect, it does improve memory in some cases. We know that acetylcholine is important to some memory. So in, for example, animal studies, acetylcholine is really important for acquisition of the fear memory. And it's also important for kind of hippocampal processing, which is your memory center. And there's a lot of evidence to suggest that acetylcholine is really important for kind of these memory processes. So again, in the human sense, it does improve memory and cognition relative to placebo. But again, we still don't know all the basic mechanisms or why. And interestingly, it also seems to delay the deposition of amyloid plaques. So it seems to kind of slow down that accumulation of those amyloid plaques. Why it does that, we're still not very sure. But this drug can also have a wide range of side effects as well, because again, acetylcholine, it's not just important for our central nervous system, acetylcholine is also really important for muscle action as well. So when you have a motor nerve that's stimulating muscle, it actually releases acetylcholine to cause muscle contraction. So if you're taking a drug that prevents the breakdown of acetylcholine, you can actually get a lot of these unwanted side effects in, for example, digestive issues, because a lot of that muscle is innervated by nerves that release acetylcholine, so you can have a lot of GI issues there. It can also affect sleep, and it can also cause headaches as well. It can also change some of the vascularity in the brain as well, which can possibly lead to headaches as well. But it does seem to improve symptoms, memory symptoms in a lot of cases. So another drug that works on the same system, but just a little bit differently, is rivastigmine. Similarly, it's a reversible inhibitor of acetylcholine esterase. but it also works on butyrylcholinesterase as well. So I didn't mention it when I was speaking, but butyrylcholinesterase also breaks down acetylcholine as well. So in this case, this drug is affecting both of those enzymes. So again, it's just increasing the amount of acetylcholine in the synapse. And again, this one seemed to be a little bit more neuroprotective. We don't know why it's a bit more neuroprotective, but it actually seems to slow the cognitive decline in Alzheimer's disease patients and actually lessen the severity of dementia. So some people have looked at, taking this a bit earlier on to kind of stop that neurodegeneration over a longer period of time. We're not really sure why it's neuroprotective or how it can be neuroprotective, but it does seem to help in the long run. But similarly, there's also a wide range of side effects as well. Digestive issues, again, similarly because you're increasing acetylcholine. And it also seems to increase aggression as well. Not really sure why. Maybe because this one affects, more of the enzyme that breaks down acetylcholine. So maybe you're getting too much acetylcholine in some regions of the brain that might be important for mood, but unsure there. And it can also cause weight loss as well. But interestingly, rivastigmine can also be used to treat Parkinson's disease dementia as well. So it can be extrapolated not just to, specifically dementia, it can also be extrapolated to Parkinson's disease dementia as well, so motor symptom dementia. So that was mostly the cholinergic system. But just like with mood disorders and anxiety, we don't always have to target, monoamines or these kind of neuromodulators. We can also affect glutamate receptors as well. So remember, glutamate's the excitatory neurotransmitter. It can bind to NMDA receptors, which cause excitation. And there's actually been some studies that show that if you inhibit glutamate excitation in some neurons, you can actually improve dementia symptoms as well. Because remember, amyloid beta accumulation affects those postsynaptic neurons and their NMDA receptors, so maybe changing that can improve some symptoms as well. And what memantine does is that it's an antagonist for NMDA receptors, so it inhibits NMDA functioning, and this reduces overstimulation of NMDA from increased glutamate. We haven't talked about it here because it's more of a neuroscience thing, but when you have neurons beginning to die in neurodegeneration, you actually see that a lot of these neurons release a lot more excitatory neurotransmitters than in a normal sense. And we call that glutamate excitotoxicity, because if you have too much glutamate in the synapse, you can actually overstimulate neurons and cause them to die. So this is kind of like this positive feedback loop. You see this in other forms of neurological diseases and disorders as well, so things like traumatic brain injury. When neurons begin to die, they release more glutamate, which then influences more neurons to become overstimulated, and that can cause their death, which causes more glutamate to be released. So it's like this positive feedback loop. And this is what happens in dementia as well. So by blocking those excitatory receptors, you can kind of reduce that overstimulation, so those neurons are less likely to be overstimulated and die from this drug. And there's not much kind of use of it in the literature, but people suggest that it may be useful in improving cognition in Alzheimer's disease and vascular dementia as well. So even though it's stopping that overstimulation, this can improve mood in some contexts as well. And sometimes this is often given in combination with those cholinergic drugs as well, because again, they work on two different systems, but they work together for a common cause. So sometimes if these acetylcholinesterase inhibitors, maybe they're working, but maybe they're not working that well, you can take memantine along with this to kind of improve those effects even more. But yeah, memantine has a very short blockade as well, so it has a very short kind of active period compared to other drugs, but it's just a consideration with it. But it is an alternative form of treatment that can actually work in combination with some of the drugs we just talked about. So what else kind of can we target for behavioral and psychological symptoms? Well, again, we talked a lot about monoamines in the previous lectures, and here, this is just a table showing you that serotonin also plays a really strong part in this behavioral and psychological regulation, so mood changes as well. So we can actually target a lot of these receptors to kind of have an antipsychotic or anti-aggressive effect as well. Even dopamine can work in some capacity, even noradrenaline. And this is also showing you some other targets. So we talked about acetylcholine, which can work on muscarinic receptors. Cannabis can actually work, or sorry, I should say endocannabinoids can also cause a therapeutic effect as well. But as you can see here, a lot of kind of these pharmacological effects can actually be brought on by the serotonergic system as well. So in some cases, again, I won't focus on this too much because we've talked about SSRIs, but small clinical studies have shown that SSRIs can treat agitation and psychosis in addition to depression in dementia patients. So you can affect a lot of different neuropsychological kind of cognitive processes with SSRIs. And these can also be less likely to cause the anticholinergic side effects of other antidepressants, because remember we talked about tricyclic antidepressants can affect multiple systems. Monoamine oxidase inhibitors can cause that cheese effect that I talked about earlier. So SSRIs might be a good kind of alternative in dementia. And they usually have good bioavailability with fairly long half-lives, so they can affect the brain for a long period of time. But there is some side effects. I didn't really talk about them in the SSRI lecture, but there is some side effects with the drugs as well. And we've already talked about this, how they work. So you've got the serotonin transporter or monoamine transporters. Neurons release serotonin. So if you have selective serotonin reuptake inhibitors, basically you stop that reuptake of serotonin, so more is present in the synapse to cause your effect. We've already talked about that. But this can be really like an alternative treatment to improve dementia-like symptoms as well as depression. And we didn't talk about it in the SSRI lecture because I didn't want to go into it too much. But a lot of these drugs, especially with SSRIs, you can change the density of the serotonin receptors. So when you take them for a long period of time, you can change the amount of serotonin synapse receptors on the postsynaptic neuron, and you can also change the serotonin receptors on the presynaptic neuron as well. But don't worry about it too much, but it's just showing you there can be alternative mechanisms for how SSRIs work as well. But it's not always the SSRIs that can cause an effect. So this is an example of a specific drug that works a little bit differently to SSRIs. So this is pimavanserin, and this is an antagonist of the 5-HT2A receptor. So this is specifically a serotonergic receptor. It's a G protein coupled receptor. So in this case, instead of blocking that serotonin transporter, the same amount of serotonin is being released, but we're just blocking specific receptors on the postsynaptic neuron. So we're just changing the activity of those receptors. And this can have a therapeutic effect as well. It seems to be selective for the 2A receptors. It also does affect the 5-HT2C subtype of the receptor, but it has more affinity for the and selectivity for the HT2A receptor. And this is FDA approved for the treatment of psychosis and hallucinations in Parkinson's disease. So it does seem to affect those psychotic symptoms. But there's also a large body of evidence investigating whether this drug can also be used to manage psychosis in dementia. Excuse me. Again, it shows promise in treating psychosis in Parkinson's. Maybe this can extend to dementia patients as well. But there's very little evidence for it, but it seems to be promising in what evidence that there is. Okay, so we talked about cholinergic drugs. We touched a little bit on glutamatergic drugs, and then we brought up the idea of SSRIs again. So these are all treatments that affect the specific neurotransmitters in the brain, right? So they're affecting excitatory neurotransmitters, all in different systems all across the brain, and they're very important for mood regulation. But this doesn't really address the issue that you do have accumulation of beta amyloid plaques and neurofibrillary tangles. Some of these drugs have promise in slowing that progression, but is there any way that we can target those specific beta amyloid plaques and neurofibrillary tangles that might have a therapeutic effect? And this is what this alternative treatment is here. So this is looking at amyloid beta antibodies, okay? Is everyone here familiar with antibodies? Maybe like immunohistochemistry, things like that? Yeah. So antibodies, they're proteins produced in our body that recognize a specific antigen. So they recognize another protein. Basically, they bind to it. And once they bind to it, you can cause a variety of different effects. You can block those proteins. You can start causing macrophages to eat the protein, things like that. So these are just proteins that recognize that amyloid beta protein. And there's actually, should not come up, but one specific amyloid beta antibody is written here, so aducanumab. I always have trouble pronouncing that, sorry. And this has actually been shown in mouse and humans to remove amyloid beta plaques in Alzheimer's patients in the phase 1 clinical trial. So this is showing here like a MRI image of the brain, and wherever you see more red, you're seeing more of the amyloid beta protein, okay? So amyloid beta aggregates. And this is in 2 individuals that have Alzheimer's disease. One individual. No, actually this is multiple individuals. So this is showing you baseline. So this is an MRI image taken before they took the drug. And then this is showing you, in this case, a placebo. But this is showing you the different doses and kind of the reduction in the amount of amyloid beta in the brain when taking this drug. So more red here at baseline. This stays the same or maybe even gets worse 1 year later in this person that had placebo. But in the people that had this amyloid beta antibody, you see that there's less red, so there's a greater reduction in amyloid beta accumulation in these brains. So it seems to specifically target this protein and stop the production of these plaques, which then might have a therapeutic effect because it might be reducing the amount of brain atrophy. It was also seen to improve clinical outcomes of cognition. So not only did it reduce amyloid beta accumulation, it also improved cognition. So it seems to improve cognitive abilities like memory and mood. And because of these promising studies, it was actually FDA accelerated approved in June 2021. So usually a phase 1 clinical trial is usually used to test the safety of drugs, and then it needs to go to phase 2 clinical trials, things like that. This can take years and years and years and millions of dollars. But because dementia is such a kind of, it's something that needs to be addressed, and there hasn't been any effective treatments, after this paper came out and was showing promise, the FDA accelerated the approval of this drug, so it actually can be used in the clinic to treat the disease. But there is a lot of controversy around that decision. So first of all, one of the big controversies, and that I mentioned before, is that is this beta amyloid actually really that bad? Again, we see increased beta amyloid in people with dementia, so we think it's a product of dementia. But there's also some studies that show that beta amyloid can actually be neuroprotective, and it might actually be the body's way of combating dementia in some sense. So if you're all of a sudden removing amyloid beta, okay, you're removing it in these people, you might be improving cognition, but maybe you might be inducing more maladaptive changes. And we're not really sure because there hasn't been that many studies looking at that. So there's one controversy surrounded about that, is kind of, is amyloid beta really that bad? But the other kind of controversy is that this drug specifically was fast-tracked. Okay, we did some phase 1 clinical trials, but a lot of people, a lot of experts in the field suggest that we need to do more clinical trials in order to understand this drug more before we start using it just blanket in people with dementia. And what adds fuel to this is that I showed you here one study that showed really promising effects. But some other studies didn't show as many promising effects. So for example, 2 phase 3 clinical trials that were using this drug were halted because a futility analysis was basically conducted. So when you're doing these clinical trials, you do things like a futility analysis. So you basically track how well patients are going with this drug. And if a lot of them aren't responding, then usually we just say we stop the study because it's futile. There's no really reason to keep going on with the study because it's not showing any therapeutic promise. So some trials showed that there was no kind of therapeutic promise in this, so it was stopped. Some of the studies showed effectiveness in lowering amyloid beta, but a lot of these wasn't actually replicated in these other studies. So even though it lowered amyloid beta in some trials it didn't do anything to amyloid beta. And in one study in particular, it actually showed that after taking this drug, there was an enlargement of the lateral ventricles in a subset of patients that were receiving this. So remember, those lateral the ventricles are those canals in your brain that are really important for insulation and kind of removing waste from the brain. But some of these people experienced an enlargement of those ventricles, which means that we're not really sure, but maybe enlargement of these ventricles might be impeding on the brain tissue, so it might be reducing the healthy brain tissue as well. But increasing the ventricles as well might also have some maladaptive effects that we just don't know yet. So not all of the findings from the studies were promising. And in the FDA, so you have kind of scientific advisors that write reports on drugs and, say, we recommend this drug to be approved, we recommend this drug not to be approved. And this is a board of like 10 to 12 scientific academics. And after this drug was approved in the FDA, I think about 10 of them resigned off the board, I think the story is, because they were just in protest of the fact that this drug just got fast-tracked when we still don't know a lot of the basic mechanisms, and some studies are showing maladaptive effects. So even though it does seem to be promising, there is some controversy in the field around that. And lastly, I just wanted to touch on a kind of non-pharmacological treatment, but can be paired with a pharmacological treatment, and that is using ultrasound stimulation in combination with other drugs like this antibody to improve the effects. So we talked about the blood-brain barrier before. So this is the vascular system in the brain, and basically it prevents certain solutes from going into the brain and going out of the brain. So it controls the amount of drugs that can get into the brain. And I mentioned that a big kind of factor in drug discovery and drug therapeutics is actually looking at ways that we can get drugs into the brain. And scanning ultrasound stimulation, or ultrasound stimulation in general, this can actually be used to open the blood-brain barrier, reversibly. So let's just say you have this ultrasound stimulation to the brain. What studies have shown is that if you use a specific frequency and do it for a specific time, you can actually create little holes in the blood-brain barrier. So you can open little pores, and then drugs or agents can then move into the brain unimpeded. They don't need that receptor-mediated transport, carrier-mediated transport, because you create a little hole in the blood-brain barrier. And then after a few minutes, or when you stop the stimulation, this blood-brain barrier just reorganizes itself again. So you can just selectively open it for short periods of time. So then if you take a drug during that period of time and then open the blood-brain barrier with this ultrasound stimulation, you can get more of the drug going into the brain. And this is what one study actually did. So it used this in a mouse model. This is the mouse brain here. This part is the hippocampus, and all these black dots that you're seeing here is amyloid beta plaques. So this is showing you in a mouse that was just so I should say this is a mouse model that has dementia. So this mouse actually develops beta amyloid plaques, which leads to dementia-like symptoms. A lot of people use this for studying drug and therapeutics in dementia. So in this mouse, you see accumulation of these plaques throughout the whole brain. You get a lot of these black dots everywhere, which is more of that protein. When you give that adubicam, which is that amyloid beta antibody, you see that there's a reduction in those black dots. So there seems to be less of those black dots. It seems to be removing that amyloid beta. If you give ultrasound stimulation alone, it also seems to improve those dots. There seems to be less amyloid beta. So just opening the blood-brain barrier for a short period of time can also have potentially a therapeutic effect, maybe more microglia going in, more immune cells, we're not sure. But when you give ultrasound stimulation and that antibody at the same time, it seems to even more remove a lot more of those plaques. So you're kind of causing more of that antibody to move into the brain, which might be clearing up those plaques even more. And this is just a graph showing it. So percentage of the plaque area, you're just seeing a reduction with those two treatments, and then an even, seems to be, well, not even a further reduction than this stimulation and drugs, seems to be a little bit of a dip compared to these other treatments. But it just goes to show you that this might be a potential therapeutic option for improving these plaques to a greater degree. And again, this is just some immunofluorescence on those same animals. So all these different colors represent different proteins that are being stained for. So DAPI is just a non-specific cell marker. So everywhere you see blue, that's just a nucleus of a cell. Doesn't have to be a neuron, just any nucleus in the brain section. Red is IBA1, so this is showing microglia. So microglia are kind of like the immune cells of the brain. This green is showing you these beta amyloids. And then this grey one here is showing you, wherever it's more kind of white, there's more of that amyloid beta antibody binding to it. So this just shows you in a normal mouse, you have microglia. These microglia are surrounding those plaques, but you don't have much of that antibody binding to those plaques. But in an animal that had ultrasound stimulation with that antibody, you see that there's less microglia around those plaques. The plaques seem to be a little bit smaller, and wherever those plaques are, there seems to be a lot of that antibody present on those same plaques. So it seems to suggest that with that stimulation, more of that antibody is seemingly getting into the brain. Whether that has more of a therapeutic effect, we don't know, but it's just something to consider as well. And this ultrasound stimulation is being used not just for dementia, but it's being investigated a lot, not just for improving mood, but a lot of like drug delivery as well. So delivering neuroprotective agents into the brain. If you can open that blood-brain barrier for a short period of time, you can increase the amount of it going into the brain for a greater effect. Anyway, that's it. So again, with dementia, again, just focus on those, how serotonergic drugs can be used, memetine, SSRIs, and then antibodies. Be aware of the controversies of amyloid beta, and also just know a little bit about the pathophysiology. So what general brain changes do we see in the brain? But that is the last lecture given by me, so thank you very much for being here these past 4 days. Dr. Alex Tang will take over the next 2 lectures. I think the next lecture's on endocannabinoids and psychotics, and then he'll finish up with photopharmacology, which is a really interesting technique. But yeah, any questions, feel free to ask them or email me. Oh, that's what I wanted to say as well. In the last lecture, I explained one of the slides a little bit incorrectly when I was talking about one of the barbiturates and benzodiazepine traces. It won't affect your interpretation of it, but any neuroscientists out there would have issue with it, so I'm going to make a quick post on the discussion board, but it won't affect the interpretation of the slide. Anyway, take care, good luck with the rest of your studies, and maybe see some of you during honours or masters talks next year.